One of the functions that glutathione has in our body is to help our proteins form these disulfide bridges. Now, a third of the proteins that we produce have these disulfide bridges, and again, it's these disulfide bridges that help the protein fold and function properly. Now, to illustrate this property that proteins have, I have an antibody that I have on the board here, again, to use as an illustration. Now, an antibody is a glycoprotein, and it is synthesized internally and then secreted either to the cell membrane or it's secreted into the surrounding environment. Now, again, an antibody is made up of both protein and carbohydrates, and it also has a lot of these disulfide bridges, or these sulfur-sulfur bonds. Now, it's these sulfur-sulfur bonds, or these disulfide bridges, that are critical for our antibodies to fold and function properly. Now, again, it's glutathione that helps our body to form these disulfide bridges. So you can see how glutathione is directly critical for proper antibody function, and then to help create a proper immune response to foreign substances, foreign particles like viruses, like COVID, like foreign bacteria. Okay, so how does an antibody work? So I'm going to go off a little from glutathione for a second, just to help illustrate how an antibody works, so you can understand the role that glutathione has in regards to these sulfur-sulfur bonds. So basically, I'm going to describe what an antibody is. And an antibody is made up of a few different segments, a few different protein segments. Now, we have what's called a heavy chain part of the antibody. Now, the heavy chain is basically what I consider the trunk of the antibody. Now, it's this part of the antibody that will bind to the membrane if it is a membrane-bound antibody. Otherwise, the antibody is secreted into the environment. So anyways, we have what's called a heavy chain, which is, I have highlighted in green, and that is again the trunk. And then we have part of the heavy chain here that is in blue. That is part of one of the arms of the antibody. Now, the heavy chain is a relatively conserved sequence, which means that it doesn't change very much. Now, when we get down to the farther parts of this arm here, we have what's called a light chain. Now, the light chain, again, is a protein segment. These are all parts of the protein, and these are incredibly variable. And it's this variability, these changing protein sequences, that allows our body to bind to antigens with such specificity. Now, what is an antigen? So you have a foreign virus or a foreign bacteria that enter your body. Now, those viruses and those foreign bacteria, they're all going to have their own proteins. Now, an antigen is usually parts of the proteins from these foreign bacteria and these viruses. Those are eligible to be antigens. Now, the ability for these light chain parts of the antibody to have such variability allows us to be able to form an immune response to these new pathogens, to these new viruses like COVID again. So an antigen, again, is part of a foreign protein that is usually part of a virus or a bacteria, and it is the light chain that are found on the far arms of the antibody that allows our body to bind to these foreign proteins with such specificity. Now, even one amino acid change in an antigen is going to determine whether the antibody will bind or not. That's the amount of specificity that these antibodies have in binding to a specific foreign particle, a specific antigen. Now, again, why do I bring this up with glutathione? Because they are loaded with these sulfur-sulfur bonds that glutathione is critical for helping us to make. You can see here, all these are sulfur-sulfur bonds. These are disulfide bonds. And again, it's the glutathione that is critical to helping us make these bonds so these antibodies can fold and function correctly. Okay, so let's go to the right side of the board quickly here. and we'll go into how we actually make these antibodies and where glutathione comes in. Now, antibodies, like most proteins, follow this same relative dogma, which is they are the genes that code for these proteins are transcripted into mRNA. The mRNA leaves the nucleus, and in this case, the mRNA, goes into what's called the rough endoplasmic reticulum. Now, again, I had mentioned that these antibodies are made up of these segments, these protein segments. So they come together in the rough endoplasmic reticulum, and this is where they are really formed into what you see on the left side of the board. And it's this glutathione that is found in the rough endoplasmic reticulum that helps create these sulfur bonds that you see on the left side of the board with the fully formed antibody. So again, the antibody is packaged and shaped in the rough endoplasmic reticulum. This is also where we add the sugar to the antibody that I do not have illustrated on the final structure. And then once that happens, it leaves the rough endoplasmic reticulum and goes to what's called a Golgi apparatus. Now, these Golgi apparatus in the rough endoplasmic reticulum, again, are just compartments in the cell that help for processing. Now, after the antibody leaves the Golgi apparatus, it is then secreted in these small packages called secretory vesicles, and at which point they can either coat the cell membrane, depending on what immune cell it is, or they can be secreted into the environment. So a B cell can produce up to 15,000 antibodies just to cover the coat of its cell membrane. Again, one B cell can have over 15,000 antibodies coating the outside of its membrane. And a plasma cell, which is another type of B cell, secretes 2,000 antibodies per second during its lifetime. Now, the plasma cells live roughly 3 days. Nevertheless, they can secrete up to 2,000 antibodies per second. And because of this, plasma cells actually have more endoplasmic reticulum than other types of cells. Okay, so I've showed you an example of disulfide bridges and how critical they are in regards to these glycoproteins antibodies. I've showed you that glutathione is critical for helping to form these disulfide bridges. So how do we raise our glutathione levels? Well, glutathione is synthesized inside the cell, which means that supplementing with glutathione really doesn't raise your glutathione levels to any appreciable level. However, whey, milk thistle, sulfur foods, N-acetylcysteine, alpha-lipoic acid, selenium, any vitamin that increases methylation, so we're talking about niacin, B6, B9, B12, biotin, vitamin C and E, liver, taurine, magnesium, SAMe, and melatonin have all been shown to raise glutathione levels. So if you're concerned about dealing with any virus, or and you want to optimize your immune system, you really want to be making sure you're eating what you need to eat to make sure you're making enough glutathione. And I've illustrated clearly how glutathione impacts the synthesis of our antibodies, which help us relieve antigens from our body. Pardon me. One closing note here: when you consider taking a supplement, you should really approach taking any supplement the same way you would with a medication, because the supplements that are available to take over the counter can have the same potency effect as any prescribed medication, or to have the same potential effect. So if you do decide to supplement with something to raise your glutathione levels, be really cognizant of the effects that you experience and be cautious with it. Okay, so I'm going to leave my sources, and I'm also going to leave all the information that I mentioned that is not on the board in the description box.